How long do you think that we'll be talking with them?
About an hour is what they blocked off, but it's whatever time you have, you know.
I hope it will be around 40 minutes, no more than that, because after that, my experience is that people get bored or something like that.
Trust me, Ron, not the community.
Nice, you're dedicated people.
They'll keep going, Ron.
Okay. I think we're good. Okay, I'm going to move you guys down, and we can start.
Okay.
Okay. All right. Hi, everybody. So this is our second AMA interview. We will... today we have Dr. Ochoa, the USDA mite expert, normally works on plant feeding mites, but also with systematics of mites and rodents in soil. He's specifically here to help us kind of understand the meaning of what the impact that mites have in our hobby and how they what the risks are really. I guess he can introduce himself more. And we also have Dr. Campbell, who is assistant professor at UNC Pembroke, and she is a specialist in mites and biodiversity, specifically in ecology and systematics. Armando, if you could help me out here.
Yes. So this was brought on because a lot of our community really doesn't know much about mites. In fact, a lot of people don't know a lot about mites. And there's this conception that most mites just are harmless, so we shouldn't really worry about them. We shouldn't care about them. There's also, now that we're opening up trade and people are going to start moving ants around more, it's important to know what you're dealing with. What is a mite? What's on there? The other thing I'll just plug in here, just as a kind of flex for Ron. Anybody remember the giant millipedes? Ron was the guy who got them banned from this country.
No way.
Oh, don't deny it now. Because of a certain mite that was on them. So this is a big deal, because these mites, if they affect our local ecology, if they affect our agricultural systems, they can end up being what regulates our ant spore.
Yeah. So. Yeah, I mean, it's as far as the hobby itself goes, and especially as smaller businesses join the hobby over time, it's really a deadly serious topic, and the more that we work with professionals in this field, the more that we understand the risks and how important it is that we pay attention to it, the better the hobby is as a whole. So that's a big reason that I was really excited to have both Dr. Campbell and Dr. Ochoa here today.
All right. And Dr. Campbell, do you have anything else to add for our intro? Otherwise, I'll let Ron get started right away with the pretty pictures. So what we're going to do is we're going to start first with a little presentation on what mites are, some of the really cool pictures we've taken here, and then we're going to go into the questions that people talked about. I'll be copy pasting the pictures into AMA voice chat.
Mm-hmm. Okay. And if we have time afterwards, if you have... We've only granted voice access to some people, but if you'd like to interject or have questions related to what's being answered, I'll be watching voice text, and the other mods will as well. And we can try and, even if you don't have voice access, we'll try and get your questions to the experts as well. So.
Dr. Campbell, Dr. Campbell, do you have any comment before I start?
I'm just excited to be here. Thanks for having me.
All right.
Okay.
Well, I'm gonna paste your first picture in there, Ron.
Yeah, we'll start with the first one,
and then you can talk about that.
Okay. As you can, I work with mites. I started to work with mites a long time ago, more than 40 years ago. They are fantastic as entomologists, and you are entomologists too, because you love to work with ants. They made you also an entomologist. They are fascinating. The persistent, the life, the resistant, the temperature, cold weather, whatever, everything that they do, they're really, really interesting. And with that, we know that mites were many, but we didn't know how many. Recently, in 2017, a group of scientists in Texas A&M did a very interesting study of biodiversity of our planet, and they discovered what was suspected a long time ago, that there are as many species as insects are. That means that there are between 5 to 11 million species of mites in the planet. Fortunately, only 60,000 of them have been described. Now, the way that we have been working with this is that we use an electron microscope called a low temperature scanning electron microscope, or cryomicroscope, where we have the ability to freeze the material. We did this with the snowflakes. We were able to freeze snowflakes. And doing that, suddenly we said, hey, one more, we can start to freeze also insects, nematodes, bacteria, fungi, whatever. Right now, we have a microscope. And with that modern microscope, we prepare our samples in copper plates. The copper plates are cut, could be a part of an insect, could be an ant with mites, could be a plant tissue, could be some water. But we put it inside liquid nitrogen at minus 192 Celsius. That is very cold. It goes freeze in seconds. From there, we store them indoors. After that, we start to look at them on microscopes. Now, here I want to stop with this photo. The USDA has been working with this ago. Our first expert on mites and insects was Pergande in 1840s to 1916, and he did some of the drawings of insects and mites that we know today. And you can see his very primitive microscope against the microscope that we use right now. Now, using cryo techniques allow us to take a picture like this one that you see right now. You see a bunch of tissue, and this tissue that you see are already captured a lot of mites feeding with the plant tissue. I know that you probably will have a hard time right now to see how many mites are, but let me help you. We do have the ability to return color, and now you can see how many mice are. And by the way, the white thing that you see there is egg. In other words, we have the ability to freeze the mite in their environment and capture their behavior. We can see if the mite is feeding, if the mite is having an association with another mite, if the mite is copulating, if the mite is dying, if the mite has been attacked, whatever the mite is doing, we are freezing that in time. One of the best things that we did was that, as working with plant-feeding mites, we work with mites that are very tiny. The smallest mite that we know is the Aerophioid mites. Aerophioid mites, like this one, is between 82 microns to 150 microns. And as you can see, that tiny little mite ability to go over the rose tissue plant and move under the trichome and feed it there, completely protected of any enemy. The red things that you see there are the globular trichomes that are supposed that the plant produced to protect herself against insects. But right now, literally, the mite not only is feeding from them, but also use the plant to protect the mite himself against predators. That shows you the ability that they have. However, to make these things more interesting also, mites have the ability to carry fungi and bacteria.
Just a second. Just a second. Is anybody else having difficulty hearing what's going on?
No, I can hear it just okay. Sounds good to me.
Okay.
Okay. As you can see in this photo, that are 82 and 100 microns in size, completely covered by fungi. And the spores do get attached, and these mites can be used in wind for dispersal. How far the mite can by a wind? It can flow in the air 600 miles, carrying the fungi or bacteria. There are other ones that are flat, and they are adapted to go over stretch, reduced areas and feeding on that. And with that, not only are able to eat, but also are learning to put the eggs in protected areas.
Keyboard is. Let's see.
Mm.
There we go.
Sorry, I just muted him.
Yeah. Okay, here you can see the egg of the mite, where the mite use the plant tissue or the material to put their eggs and protect it. Keep this in mind too, because this will happen in your colonies, where the different mites will put the eggs in different parts that you are not aware, and maybe will looks normal, maybe part of the ecosystem, maybe part of nets, maybe part of the soil that you are using as a media. Other quality that mites have also is their tegument, their skin, to be particular. Right now, I will show you a mite. It's another flat mite, again, associated with plants. What was interesting about this mite is tegument. Tegument is produced by some pores over the body, and it will spread all over the body. And it's a kind of a wax-like substance. However, we discover that in reality is protein. That shows you that allow them to be more resistant to cold or heat. And some of them are beautiful. Some mites that you will encounter can have very beautiful colors. But however, other mites will be kind of plain color. And with that, it can camouflage also with the background, like this one from Australia that you will see right now. And what also interesting is that the legs of the mites have a very sophisticated claw systems that allow them to grab and move, not grab and move, but also to run. I don't know if you are aware that one of the...
So just something to clear up real
Yeah.
quick, Ron.
Yeah.
You and Gary Bouchon, who take these pictures, you have a technique to capture the color.
Yeah, I will mention that later.
Okay,
I will mention it later. When we finish,
they had a bit of a question wondering on SEM black and white, so how are these colored?
Yeah, we have a technique to that, and we'll explain to them after. Okay, look at this leg of this mite that you are looking now. I don't know if you are aware that recently search of somebody that tried to check how fast mites run, and they discovered that if it's true that the cheetah can go 60 miles per hour really quick, really fast, when they check the mites, they found that there are mites that they run close to or more than 300 miles per hour in proportion. That show you that the mite can be inside your ant colony, running from one side to the other side, and a hard time keep it where the mite is and where the mite goes. Another character of the mites is the cities that you probably will call hairs, and the hairs are modified to protect also, to absorb humidity, to protect against predators, to protect them against substance. One of the most common mites that you are aware is the two-spotted spider mite is the most common one, and probably you will see that they produce webbing and all that. That is easy to see. However, introduce you now to details. For example, these are the eyes. The first eye allow them to see contrast between dark and light. second,
really quickly, Armando, could you set it up so that it's push to talk, so that we can just hold the button down? Is that possible?
Yeah, let me see. I don't know if it's that or if it's, whenever he's talking, it's chewing up all the bandwidth. I mean, whenever I'm uploading.
Okay.
Let me
that could
check.
be it.
Yeah, because it seems to happen every single doing that.
I see. Okay.
okay.
All right. Yeah, we can just continue. It's fine.
Okay, we'll continue it as is.
What is okay? And here,
Because it's getting choppy whenever I upload something.
oh, okay.
You see that little signal bar at the bottom?
Okay, that means that your computer is not good enough.
Internet connection is not good enough because we're not doing this in my house.
Okay. In this one, you can see the eyes of the mite, how sophisticated are. What I am pointing to you with this photo of the eyes is that show you that mites have better way to be aware of the surroundings, much better than probably any of us. And now the next one will be the mouse parts. With the mouse parts, look how complicated the mouse parts are and how intrinsic are. And many mice have different types of mouse parts. Some will have a stylet way, some ones will have a chelicera, some ones will be having a mix of broken chelicera that allow it to cut and hold or suck aliments, food, and other things. With that, let me introduce you now about the problem when you are working with communities, be insects, be reptiles, be any pet that you want. Okay, ready? What you see right now is a gecko skin. Geckos are very popular. Now go to the next one.
Let's see if we can upload that one. There we go.
Geckos are very popular, and the people start to bring them. Some people did bring geckos illegal, and the geckos came with a phoretic mite because was carrying on it. But this phoretic mite was not only phoretic, this mite was making holes in the tegument, feeding and taking out blood of the geckos. Now, the African gecko didn't have too much problem because has been living with this phoretic parasitic mite for centuries. But with the gecko from Africa was introduced to the United States, these mice start to jump from the African lizard to the American lizards. The American lizards were not exposed to these mites, and you can guess what happened. They start to die because they are not tolerant or accustomed to this kind of association. And that is the concerns that we have with. Let me help you a little more. We humans have the similar situation. And with what we know about that is with ticks. Ticks are mites too. And ticks are able to be phoretic, moving in other organisms, but at the same time, the mouth parts to parasite us. And it can give us diseases, rickettsias, Lyme disease, Rocky Mountain spot fever, hard water disease, all these things. Again, this is association between a tick and a human. All right. With that, what happened when we use soil? All that we will encounter a different world, the world of mice associated with. Some of them are beneficial because they will process and eat the soil, filter the soil, move around. But some of them also sometimes they can carry the parasite inside that can affect mammals and things like that. Oribates are very good mites, are beneficial, and they are common to be encountered in soil and in colonies. Next.
Just so everybody knows, the discussion and the photos that are being talked about are being sent in the voice text channel. If anybody was missing it.
Okay, thank you. Now, I will educate you today with something very. Every day we hear about people suffering allergies with dust mites. The mite that you will see now It's okay, Ian?
You want me to put it in?
Yeah, please.
All right.
The mite that you will see is the true dust mite. Please look at it because if you see only have 2 pairs of long hairs at the end or 2 pairs of long city at the end, that is the dust mite. And you are allergic not to the mite, but you are allergic to the poo of the mite because the mite feed on your dead skin. Now, this mite, be aware it because this mite will be associated but not with colonies. However, the next mite,
See if we can up. Why is it doing that? There we go. Now it's uploading.
Yeah.
Right?
the next mite,
It's gonna lower the connection. Like
yeah, okay, this looks similar to your dust mite. This mite is the mold mite. The scientific name is Tyrophagus prunescenti. This mite carry fungi, carry bacteria. They will eat anything that die or is organic. It will be, if you are not careful with colonies, if you are not careful with material, clean your plastic containers, if you're not clean the little soil that you bring or a little stick that you want to put on the area where you have your ants, it will get in and will contaminate your colony. It will be going to millions really quick and annoy your ants. And perhaps eat your ants too. That means that cleaning is something important, especially with mold mite. And again, if the mite does not do that directly, remember it carries fungi and it carries bacteria that can be harmless or can be harmful to your ants. Now, here is another one. That is a smiley chigger. As you can see, the nice eyes and the smile. That mite is not associated with the ants, it's associated with get by chiggers. However, there is another mite very similar to that one that is red and runs all around really fast. You can see them in your homes and outside. There are predators that are good. However, I don't know if you want to have a mite like that because a mite like that can also fit in the eggs of small ants. Okay.
There we go.
Okay. Now, please now put attention to this photo. There is a caterpillar there that is completely in it's not completely dead, it's paralyzed by one bite of the mite. This mite that you see there is called the itch mite. The itch mite is called Pymotes, Pymotes. This is your most dangerous mortal enemy in any ant colony. If your ant colony get contaminated by this mite, it will be killed totally, completely. Each female can produce 120 to 140 new females.
1 second. So the photo that because this is going to be transcribed, the photo that he's referring to is the one with the caterpillar. Yeah, okay, specific.
Yeah. And by the way, this mite, Pymotes, has been used in experiments as a biocontrol of ants. And again, all depends how clean and how careful are you with the time that you collect your material. A queen can take out with one of these mites and in confining conditions will kill your colony. All right.
Would those be visible on the queen?
Would they be able to see them?
If you have, they can buy a magnified visor.
Yeah, the ones that jewelers use.
Yeah, you can use, hopefully give you 25x, and they can see it. When the mite is pregnant, it can grow 20 times her size. And you can see in the photo over the caterpillars that some already are bigger than other ones because they are pregnant. Okay, the next one. This is a very interesting mite, and you had two photos because you close your eyes see in 3D. The photos that try to help you. This is a
really quickly. One of our members asked if we do see those mites, the ones that would kill the entire colony, how would we stop it?
Destroy the colony, clean with Clorox, and hope there's nothing left.
Okay. So it's
Brimstone.
there's no in between. So if we see those mites, we just have to.
It's not between. You not play games with Pimotes. Pimotes can also bite the owner of the colony and give a itch system. It's called the disease is called acariosis. The mite that produces acariosis sometimes in humans. It's associated with store grains. It's a pest. It's very hard to cleaning to be clean and prevent is the best tool against that mite.
Okay. So it's not one of those things where we can like very carefully try and like scrape it off. It just we have to completely.
You don't. Furthermore, when I work with that mite, I use gloves and sometimes I bite.
Yeah. Okay. Wow.
It's a mite that really can destroy your colony.
Okay.
And if you have several colonies in your home, you cannot risk that with that mite.
Understood.
Yeah, Pimotes is a very tiny little and can be moved just by. Okay, Ascutacaridae, this is one of the most interesting ones. As you can see, it looks like a little turtle. Maybe Kathleen can add something about them. They are really cute, really nice. Usually they are grabbing the antenna of the ants. And in base of the literature, they are considered phoretic, but also parasitoid. Parasitoid means that it will kill the host. We not know too much. Your group will have a unique opportunity to learn about them and help us to understand better. This mite, the larvae of this mite, is similar in shape and form and color to the larvae of the ants.
Wow.
Yeah. Okay.
Hey, Ron.
Yeah.
Yeah, this is one of the ones that I find the most on the ants that I was working with in Ohio, where I originally was working. And I find them on fire ants. And I don't know, I haven't seen them actively doing anything on the ants, so I'm not sure if they're the really bad parasitoid kinds, but this is a super diverse group and very, very common.
Yeah. And that is and because that is important observations to help us to understand the community working with ants can help to understand these associations. Thank you. Okay. Next one.
It's uploading. It's eating up all the bandwidth again.
Okay. Here is a very nice association between a mite and an insect that all of you know well. This is the tracheal bee mite. And you can see how the mite have the ability to go inside the respiratory system of the bee, completely conglomerated and under contained situations, especially in winter, the population inside the respiratory tracts will grow to the level that start to kill the bees. Okay, in ant colonies, you are confining your ants to a stretched area where there is no movement of the ants outdoors, outside to clean themselves and propagate it. And think about if you happen to be that have a colony with mites start to grow and grow and grow. That is the reason that I enclosed to you a little paper that we published a long time ago with a similar mite growing in association with beetles under confined conditions. And we were able to prove that the mites will grow so many that will attach to all around the mouth parts of the insects. And with that, the insect will not be able to feed and die by starvation. The next one is another association. This is with beetles. In this one, a nematode is part of the associations. Ants carry nematodes. And with that, we have another thing that we know very little. What is the association between and why nematodes and mice are sometimes together? Sometimes the mice are good. Sometimes you will have mice that are good with the ants, helping the ants to kill nematodes. Sometimes you have nematodes that kill the ants, and vice versa. And we're still learning. Next.
So you're saying that other than I mean, a lot of the time in the ant keeping hobby, when our colonies die, we don't know what's going on. They either they stop feeding, they stop taking food, or I mean, there's all sorts of ways that our colonies just die. You're saying that other than mites, there's nematodes and other parasitoids that it's not even on our radar. Okay.
That's right. And some of them can be carried by the mite.
Okay.
The mite can carry a nematode, the mite can carry fungi, the mite can carry a bacteria. When you lose a colony, you should at least take a time to look with a magnifier if you see something on the body of the ants, if you see something on the food of the ants, if you see something associated with fungi, because some mites will go over the fungi resources of the ant that is feeding the ants and bring a different fungi that will start to eat the fungi that the ant is eating.
Wow.
And affect the ants too.
That's really great information. That's not something that's been known at all in the hobby, at all.
Still a lot of unknowns.
Exactly.
And here, the next one, for example, here mosquitoes. How many times we have problems with mosquitoes? However, the mosquitoes that happen to be mites, and look how well adapted are to parasite this mosquito, this tiger mosquito. And again, the ants will go with that. We are getting close now to the ants. One thing I want to show you also is predator mite. This is the chelicera of a predator mite. This predator mite had the ability to cut a collembola. I don't know if you are aware. Springtails. And springtails sometimes are associated and all that. And this mite had the ability to cut in half a collembola. And that is very impressive.
Wow. Yeah, okay. Yeah. So just as an extrapolation of what you're positing here, a lot of the time for a lot of ant keepers, what we'll do is we will create a bioactive terrarium for the colony for them to, and we will introduce stuff like springtails to the outworld, and we'll introduce other kinds of insects. So what you're suggesting is that even for those insects that we introduce, we should be going over them very carefully, because at this level, at this very, we could potentially be introducing something that could be killing our colonies.
Yeah, exactly. You are correct. What I am doing is to bring you. Here is the beauty of this. It's not that you are getting introduced to the ant world. What you are doing is getting introduced to a micro that the ants can see, but you not. I mean that you need to learn to see it, to prevent it. For example, the next one is already done. This is one of my favorite photos. So, there you go. This photo for me is a very important photo because this was the first time that we used the cryo technique to study an association between an insect and mite. And happened to be that was a termite. And when we were looking was what these mites are doing to the termite. As you know, termites is a problem in Louisiana and other areas, which affect us and cost us millions of dollars in losses because treatment for the wood. Okay, the termite was already attached with all these ants. Sorry, the termite was attached with all these mites. And we were looking at the mites and trying to understand, okay, this is phoretic, this is not doing anything, whatever. And happened that there was a paper by Dr. Hook from University of Arizona where she was able to study this association of these mites attached to the termite tegument, and they do not have mouse parts, They are only phoretic. They are supposed that they are not doing anything. However, when we remove the mite that was attached to the tegument, we were able to see depressions caused by the anus of the mite, where was extracting humidity, water of the tegument of the host.
Wow.
And that means that is causing a stress, not only to the tegument capability of the insect, but also taking away water from the host. That's a really cool stuff.
Yeah, that's crazy. Holy cow.
Yeah, this is really cool because if you think about that, you're feeding with your butt. Well, yeah, how many of you were ready for this? Okay, let's go to the ant.
Oh wait, that's the wrong picture. Hold on, I
Yeah,
uploaded the wrong one.
yeah, you're on the we'll give you two versions of this photo because the first one is more or less what you see, and the second one is to help to see what is going on. Usually, when we are looking at, they will have similar colors in red, brown, or yellow, brown colors. I am pointing there the associations. You can see the nice ant with several mites, and some of the mites have mites, and some of the mites have a nematode, and all that. And you can see how well are adapted. The mite that is attached to the head of the ant have the ability to talk with the other ant when it comes, and induce the other ant that is giving the food by trophallaxis to give the food instead to the ant, give the food to the mite, stealing the food from the mouth of the host. Kathleen, maybe you want to say something about this because this is one of your material.
Yeah, this is an ant that I sent along with Ron that I knew had a bunch of mites on it at the time. And this is Lasius umbratus is the species of ant. And the mite, the really big mite, is called Antonophorus. So if you're interested in that, it's really cool. They use those long front legs to tap the mouth parts of their host and also neighboring ants. Like you see, there's actually another Antonophorus on the abdomen, and you can see its little legs is kind of riding on the side with the blue arrow. And so that one is probably tapping neighboring ants that are walking by. And it makes it really hard for the ants to do their job. So they, spend a bunch of time trying to get the mite off of their heads, and then they're spending much time feeding the mite instead of the larvae that they're supposed to be taking care of.
Mm-hmm.
Yeah. And the white mites, in the white mites, there is not one species. You have three different species of white mites there. You have Histiostomatidae, Acaridae, and one of them have a little nematode over the dorsum. And we still not know exactly what is the interaction between all that, right, Kathleen?
Yeah, that's correct. That's the first time that since when you took this picture, I had no idea that the nematodes were also riding on the mites. And, with a regular stereoscope, you probably wouldn't see them. So this is something really special that excited me.
Yeah, so what you're showing us here is you're showing us an ant that has a mite, and the mite has mites, and then the mites that's on the mite has nematodes.
Yeah. And the nematode is yellow color there for you to be able to see.
Wow.
Yeah, it's the one that's on the abdomen. It's got, the mite on
okay.
With the nematode on its back.
No wonder the ants can survive it all.
Oh, yeah. No, that is a good point because ants also are resilient. Ants are extraordinary colony. Remember that ants are one of the most well-organized organisms in this planet. And that is the good thing for you guys. The ants have workers, have nurses, have soldiers. They have amazing, well-organized colony. And can tell you when something is going wrong. You need to learn to read the ant behavior, especially when they are associated with mites.
Could you elaborate on that a bit? What do you mean by read the ant behavior when they have mites?
Well, like you have a dog. When you have a dog, you know when the dog is sad, when the dog needs food, when the dog is having hydration, missing water problems, when the dog needs space. It's the same with the ants. It's a well-known behavior of the ants.
So-
There is a
Yeah.
big book by my gosh, Kathleen, big book. I forgot the
Big
name.
book.
Wilson.
Wilson.
Oh yeah, E.O. Wilson, the ants. If you don't have it and you're studying ants, you need that.
Yeah.
Yes. Yeah, and that I recommended. I recommended that you get a copy of the Wilson book. I hope it's not too expensive. And I recommended to get the manual of crans for the mites and be familiar with your ant colony.
Yeah, another thing with that, with their behavior, like when I've seen them super loaded with mites, they're just kind of stumbling around. There's mites on their eyes. They're not doing their normal behaviors. They're not following the trails well. So you might notice that your colony's suffering. You might notice more of them are dead. So
Wow.
that's kind of ways that you can gauge their behavior.
It sounds like should be a wiki somewhere where someone's going to start a log and people who are keeping ants can record their observations.
Yes.
Yeah, that's one of our
Great
goals.
idea.
So
That will help us because again the problem is that this movement of colonies of ants and collection of ants are happening so fast, all of us, that really we need the help of the community to understand what is going on. In the way that you help us, we can help you more to understand what is going on.
exactly.
Let's go over the next photo because here is another thing. Because I don't want to say that all
Brian,
the
you said it.
okay, thank you. Yeah, I want to go fast.
I mean, you can stay longer
no,
for the
okay.
questions.
Yeah, we've got tons of questions for you as we're going.
Yeah.
Like especially when you said that the community needs to help you. People really want to know, like if you can the more we have more information that you can provide on behavior for ants that have mites, especially mites that we can't see. So if we can determine that we should be looking into our colony to see what kind of mites they might have and how we can send those specimens along to you. And like how it helps you, like what kind of stuff, because we've already started that process a little bit with Zanuri in our community. He sent mites along and caused a big hubbub over there apparently. So I mean, if you could speak to that a little bit before continuing, people are very interested. We're getting a lot of questions on that.
Okay, Kathleen, you first and I second.
Okay. I think what would be really nice is if people could take pictures of things and describe their observations of what they're seeing, understanding some of the associations between the different like species is hard because you have to be able to identify the mites and the ants, right? So those would probably have to be sent to a specialist. And while I am happy to accept some, I can't accept 2,400 people's mites from their ants. So periodically people have sent me things and it takes me a very long time to get back to them because I do have heavy teaching responsibilities, but
Sure.
I could try.
What we could do specifically is if you can we have a bunch of people in the community that have like focus stacking setups. If you can, and we have like tutorials on how to do that kind of thing. What we can do just to make sure that the good things get to you is we can have like kind of a vetting process. So somebody says in the community, hey, I think my ant has mites, and then what I would do is I would create like a group of people that would be like specialists and like be able to like they would have like rudimentary training in this kind of thing, and they would send the photo to them and they would vet that this is something that should be sent along to you and escalate it through different people. I mean, that's something that we could do easily.
That could make it more manageable.
Yeah, exactly. That's the hope. So
Yeah, for example, as you can see already, so people are sending photos of ants with the mites, and that will help us. But also, again, the key here for you, when you are acquiring the ant, you need to know where that ant is from. Who sent it to you? What is the origin of that ant? What is the humidity and the environment of the ant? And you are putting that all together. That is the key. Collector, place, and locality, all that helps.
That's really interesting because when something that's known for the anters in the community, anters being people who go out to the nuptial flights and collect queens, something that's known to the more experienced anters is in certain locations where we find certain species, virtually all of the ants that we collect in the area will have like specific types of mites. Just they'll be covered in them, and I feel like that kind of information passed along to you guys could be really interesting and helpful.
Yeah.
Okay.
Actually, I did a study on mites that were on the alates on Mesopurgandii in Arizona. And I found that the mites were way more frequent on the female alates than the male alates. So they're trying to get to a place where they're going to establish a nest.
Yeah. And that kind of information, that kind of records is what help us.
Okay.
And I've sent a bunch of stuff in to Ron even more recently that he probably will get on Monday.
Yeah, Michelle has it, Ron.
So,
We will look at them.
But the one that I just posted a picture of with that mite on the ant head is the one that caused all the hubbub.
What is that ant?
Yeah,
that is a Crematagaster queen, and they had 4 of those on her, and currently I do believe that it has been completely unidentified.
I think Ron got it to family, but I tossed it in the electron microscope. I don't know that I have the pictures on hand.
Yeah, we send this specimen also because here is another thing that you understand. Kathleen and I are just 2 mite people working with them, but like I said, 5 to 11 million species of mites is a lot of mites to handle. But we are lucky. We have another very important and amazing mite expert that is Dr. Hans Copeland, and he's working with the Uropodina group, and he is looking at it. What we will do is I usually will filter the material. I organize the material that only send him material that he is able to work with molecular techniques and microscopy to try to understand the role and why is the mite doing, why is there, what is the mite looking for. And that is what we want to do. And here, by the way, there are the photos of the mite that you sent us with tabletop SCM.
Yeah, there's a picture of the. I think you guys have a picture of the mouth parts as well.
Yeah, I see with the close up
somewhere in there.
Yeah, somewhere.
I can find it.
What was your identification on that, Ron?
It was a.
I don't remember. You wrote it on the phone.
Yeah, I wrote it. Mine are that sometimes are more complicated than insects because the people that work with insects took all the nice names and left us with but it's a family to something with T. I forgot that one. It's a Mesostigmata.
Yeah.
Uropodina Mesostigmata, but I think we have a possible genus, and that is what I am working Hans.
Yeah, Hans is the expert on that group.
Yeah. And by the way, we only know 100 specimens from the United States of this group. Yeah. By the way, I put a picture before. I didn't talk about. It is important to realize also not of this ecosystem between ants and mites. The ant can carry a bad mite over her body, but also the ant can carry a good mite that the ant needs to help her to have this predator mite moving around her body, killing the bad mites that she also can get. And that is the beauty of the ecosystem. Now, the problem is when you confine your colony, how I can handle that confined colony that I balance that. I also have another photo. Did you put another photo? Yeah, put another photo. I have another photo of
questions eventually.
A predator. Sorry, it's the last one. I put a photo of a predator just to look how massive the predator can be and how strong that chelicera can be to attack and kill other mites. And with that, I see that. Let me see, what is that?
That's Varroa.
Okay, and with that, I want to tell you a surprise because all of you working with ants should understand how difficult it is for us to work with mice because the size. Right now, you're looking at a photo of Varroa. Varroa is the mite that parasite bees for 63 years because as scientists in Russia, they were saying that Varroa was phoretic on the bees and they do nothing on the adults, only the larvae. And that was what everybody said. Oh, it's just phoretic on the adults. Do not harm the bee. We were able in this past year to prove that Varroa was phoretic on the bee, but also parasitic, and there was directly feeding the fat from the abdomen of the adult bee. And that bee eventually in winter, because the mite took away the fat for energy, will die.
Okay.
Okay, and with that, I end my presentation. Ready for questions?
Okay.
I'm gonna.
Yeah, put that.
And I want to put the disclaimer in anyway.
Yeah.
We are USDA scientists, but these views do not represent the USDA or the United States government. And I'm going to put my disclaimer in there as well.
Okay.
Okay.
Dr. Campbell, did you want to turn to speak on your specialty or any insight that you can share with us before we go into questions?
I'd like to just get to the questions.
Okay. Yeah. All right. Great. Okay. So I will be reading the questions. Okay. So first,
sorry.
Go ahead.
I forgot the color. How we do SEMs in color. Before we put the material inside the SEM, we will put the material alive under a high resolution Hyrox microscope. We capture the color, store that data in Microsoft, and when we go the black and white, the computer will return the color. That's the way that we do it.
Okay.
Now, the only limitation is that remember that the computers cannot do the contrast. That is the reason that looks a little artificial, but eventually the process will. That's it.
Okay.
Okay, sorry.
No, it's all good. Okay, so Armjon asks, how do you identify if a mite is harmful to your ants or if it's just chilling there?
Observation.
Okay.
You need to start to observe.
If you got them identified, then you could tell based on what others do in that genus if it's known. So many of these are not described species or totally new species to science, but you could kind of maybe tell if that's known to be a predatory genus or a parasitic genus, what it might be doing.
I see. Okay. So so I'd say we see mites about once a week in our community. Somebody brings it up. Should we be forwarding those to USDA? Or
okay, in that case, I think that need to be somebody that be like a common coordinator of that, because once that is identified one, you don't want to see that over and over at the same.
Understood.
Yeah, the reason that I showed you the photo of Tyrophagus protisentii, that one that I hope you have it and go back, is the yellow mite, the mole mite. I show you that because 80% of the material that we receive on houses and storerooms and things like that happen to be that mite, and that mite is a contaminant and is associated with how clean are you. If you vacuum your home, if you vacuum the sofa, under the refrigerator, if you wash your pillows and cover pillows and all that, if you do all that, you will not have that mite. But if you don't, trust me, that mite is there, very happy. And it's very common, and it's the most common contaminant. And I saw a couple of photos, several years, of ant colonies with this mite. And it's a contamination. It's a contamination of material, contamination of the mouth, the hands, whatever you are putting it.
And Ron, is it's immature stage? Is it phoretic?
No, they do not have a phoretic stage, but if you have some glucose for the ants and things like that, that mite would be happy and having tons of babies. All right. Yeah, it's there, the yellow. Yeah, go.
Okay.
Go on.
If a mite has... So this is something that since people have been excited and kind of talking about mites a lot, this is something that has come up. I guess we can just kind of start with how this is tangentially related to. So in the hobby, so let's just set the record straight on this. In the hobby, it has been traditionally suggested and is to use lemon juice. So can we put that to rest? There's absolutely nothing that lemon juice will do, correct?
I think that it just needs to be really tested. It hasn't ever been scientifically tested. It's people just, it's like, wives' tales sort of health treatments that humans have. How do we really know unless we have like a control group and an experimental group and we really test it? Yeah. Has anybody done that?
Yeah. And another thing, remember, we are talking about acid that burns bacteria and burns things in food, all that. And I will say, yeah, maybe it's burning the mite, but also it's burning the ant.
Well, the way that ant keepers will typically do this is they won't just put the lemon juice like directly on the ants to get them to fall off. What they'll do is they'll put lemon in the outworld and they'll let the ants kind of like interact with it. And that's, I guess sometimes people see improvements in that regard.
Yeah, I think I will go with what Kathleen said. They need to be more careful documented. Yeah. They need to have the correct dosification of how much lemon put on the water and all that.
Yeah, have before and after measurements of the mites.
Yeah.
Okay. Another thing, because somebody had asked me about that for treating them, and I said I really didn't know, but I was thinking about honeybees and how they use oxalic acid and formic acid. Formic acid, is something that's produced by the ants. Yeah. And so they use those to treat varroa mites, which are a major pest. So it's possible that the acids could, harm the mites while not harming the ants enough to kill them.
Wow.
Yeah. And another thing also, and Kathleen can help me with this. Also, remember that oils tends to cover a tiny little mite. Oil will cover the surface, and the mites have only 2 spiracles to breathe. If these spiracles are covered, you are affecting the mite.
Huh.
Yeah, but also, ants have spiracles. You wouldn't want to get them too oily either, so it'd have to be something in between.
Yeah, in between, that's right.
Okay. The other thing that has been suggested in the community, and this is what the question is, if a mite has been identified as harmful, is it a good idea to introduce predatory mites to try to control the harmful ones? And if so, where can you get them?
Predator mites are sold everywhere. You can buy Phytoceolus Neoceolus cucumeris. There are a lot of predator mites that can be bought in the market, especially in California, and you just write predator mites and you can buy them. The thing that is important here is see what could show up more associate. And I know that several of these predator mites will eat the mole mites. Perhaps will eat some of the astigmata mites because are similar in body and texture than the mole mites, and that is something that can be tested.
Okay, all right, that's something for our community.
How many of all that all depends on the size of your colony, and we don't have too much information.
Okay.
Would they be harmful to the colony is, I think, the big question that people ask too.
The Phytoceidae perhaps can give a little bite to the larvae of the ants to get humidity, but usually it's not something documented. And one thing is that some ants that you are working on.
Sorry, you cut out in the middle of that.
Okay. There are some ants that use scales to get sugar things and something like that, and some predator mites will eat the scales, and the ants will try to eliminate the mites because they don't want their scales to be affected.
So these are insects kind of like the aphids that ants tend.
Yeah.
Producing honeydew, and that's what he's talking about.
Oh, I see. Okay.
Mesostick, probably associated.
This is really insightful for a ton of reasons. It's really amazing. Okay.
There is a poster also that
I'll put the poster on.
Yeah, there is a poster. Okay. Kathleen, what you pronounce his last name? Rettenmeier?
Rettenmeier, yeah.
Yeah, Rettenmeier was amazing, and my lover. He did know a lot. Unfortunately, he passed away several years ago, but he did a lot of research on this. It will be really good to get more familiar with all these things that he did. Again, I remember that he always said, we need more people looking at, getting notes, helping. And again, be careful your colonies. Don't be promiscuous, passing colonies to one friend to another friend without making sure that you are not passing a problem.
Mm-hmm.
Yeah.
Okay.
I've also a lot of times I'll get emails from people when suddenly the mites have gone crazy on their ants, and it those mites were there the whole time probably, and then it's some change in the humidity or the food or something. The temperature has caused them to go into that phoretic stage. So the phoretic stage is just a life stage. It's like a immature stage, and so the adults might have been just eating the food or hanging out in the soil part of your nest. But then they get on the ants when something seems like it's going downhill in your colony. And then people notice that their colony goes downhill rapidly, and it could be because of the mites, or it could be because of the humidity problems or temperature of the colony.
Mm.
Yeah. For example, the paper that I sent you, Lartoglyphus, that is the mite associated with the beetles that are used in museums to clean bombs. The phoretic mite was not doing any damage to the caterpillar, but there were so many that the caterpillar no longer can eat it. And we start to observe, okay, what is going on? We start to observe the caterpillar suddenly die, and the phoretic mice molt to adults and start to eat the caterpillar. That means that what the phoretic mite was doing was waiting for his host just to die.
You cut out again.
Well, I think there's a few species that hang out.
Hang out and wait for the host to die,
and then they eat them.
Yeah, that's right. And then they molt and they eat them.
Wow.
It's like you carry with you the organisms that will eat you eventually when you die.
Jeez. Okay, next question. What? Just a second. Okay, what causes mites to show up in ant colonies, and are there ways to prevent this without having detrimental impacts on the colony?
I think that they're brought in from the start most of the time, or they're brought in on food. And so one way that I know that people have kept probably most people in this group know about ways to keep the, contaminants out of your colony. But one way that I've heard is that people have boiled the mealworms or frozen them for a long time before introducing them for food, or whatever crickets, whatever you're feeding your colony. And that way you're going to reduce the chances that you're going to be introducing the mites through the food. But I think a lot of them are on the queens to start off. That's their first chance to get into a nest. And a lot of these are associated with specific species, but not all of them. So they could transfer between species too.
I see.
If you see the 3 pictures I posted, then you'll see just about 3 weeks ago, we had nuptial flights here for C. fragilis in Arizona. And I don't know, that looks more like a tick to me, but and I actually sent these in.
Yeah, these are
in your hands, Ron.
Yeah,
those are definitely mites. Yeah.
Oh, they are mites. Okay.
Mm-hmm.
So I had to pick these off. I think the vial that I sent you has either 16 or 17 of these in it.
Yeah. Thank you very much. Thank you. One thing, for example, when you decide to do your colony, the first thing that you need to be aware is that your container is clean. Second, make sure that you have your hands clean. Next, what kind of soil or material are you putting in the colony that is not contaminated? One easy way to contaminate your home is the dog food. The dog food sometimes is easy environment for have some of these mice that when you have a container for ants, it's the same mite and things like that.
Wow. Okay. So even on like prepackaged food, so some people give their ants like fish food and stuff. Even prepackaged food could be contaminated.
Yeah.
Yeah, that's why they have sell-by dates on a lot of things like your cereal or whatever, because there's eggs in there that can hatch later on down the road.
Wow.
Yeah.
That's something that I could have lived my whole life without knowing.
Yeah. Some mites have the amazing ability, depends on the temperature, to put two kinds of eggs. Sometimes the egg is in dipause, and sometimes the egg is latent. The egg that is latent will take 1 to 3 days or 8 days to hatch. The egg that is in dipause will need a low temperature going up, like you go to let's say 30 Fahrenheit, and suddenly it goes a spring of 40, 50, and the egg will hatch after that, but not before. That means that egg can be in an egg for months.
Yeah. Okay. Chad's kind of freaking out about it.
I'm sorry about the cereal.
I can tell you, like, one of the mites I encountered that is in Ron's hands as of, I think, the 12th. He hasn't gotten it yet, but I'm gonna post a picture here of this queen that I attempted to rid the mites by putting her in a refrigerator and causing diapause or whatever, but it didn't work. The mites actually were able to survive the cold even longer than the queen could.
Yeah,
so
that's what I've noticed too, because I would take pictures of the ants with the mites by putting them on like ice, kind of on like a cold dish, and the mites would just be moving around just fine, but the ants would kind of stop pretty quickly.
Yeah,
I'm trying to find. Oh, here it is.
Yeah, same thing like with breathing and things like that. The ants are gonna stop breathing before the mites would.
So this is a Nova Messer Coccarelli queen, and she was literally covered in all of those mites. That's just her gaster from one angle, but it was probably I would say triple that number there that you can see of the mites. I could not get them all off, so I basically sacrificed her for science and stuck her in a ethyl alcohol vial and sent the mites and the host to Ron. So those are there with you.
Yeah, it's always good to get the host with the mites. So I noticed some people were talking about whether they should just send the mites or not. And one reason why is because when people try to transfer them into a vial, the mites get lost pretty easily. It's really easy to flick them to somewhere and never find them again. And also so that we can have the reference voucher specimen with it so we know the true host, and we'll keep that identification with it always.
Okay.
By the way, talking about how they send the specimens and things like that, you can use a small vial with just 1 cc of 70% alcohol and put it inside, tie it well, put some kind of wrapper around the top that the alcohol will not come off, put it inside a plastic bag, close the plastic bag, put the data with pencil that do not run off in case that the alcohol come out. And you can mail it in these envelopes that have cushion inside, and in that way it will be the safety way to mail.
Yeah, and I've also heard you should probably put some paper towels in there. Should it leak, the U.S. mail or whoever mail isn't going to be super happy to find a leaky package of potentially flammable liquids. So keep it in a plastic tube that you're going to send it in, and then wrap that really tight with saran wrap or plastic wrap or something so it doesn't leak. Put some paper towels in the Ziploc bag and then put it in a soft mailer.
Yeah, use small vials, tight small vials. There are tiny small vials that only hold the size of the ants, and that will be the best for us and for everybody.
Yeah, it's really hard to find them if they're in a big vial.
I actually just posted a link in the text chat for the vials that I've been using to send to Ron.
Yeah, those are perfect. Yeah.
That is a really good one, by the way.
Yeah, no taller than an inch. Yeah, that's good.
Okay. The next question is, can all ant colonies get mites, or is it something that's much more common in certain species?
So I think that all ants have mites, in my personal opinion, but the size of the ant makes a big difference. So one of my studies that I did was looking at, why do some ants have more mites than others? And so, for example, carpenter ants, much larger ants, have a lot more mites than tiny ants. It also depends on how often they move their nests around. So some species are known for moving their nests, like Taponoma ants and Solenopsis. They can raft and move off somewhere if they get flooded out. So those ones tend to have a lot less mites because they'll abandon some of the stages back, like the adult stages that aren't phoretic, back at the old place they were living. Also, I've noticed that laziest species, especially the social parasitic types, they have a lot more because they're picking up the mites that were in the colony that they invaded. That's where the queen of the social parasite comes into the not social parasite species. And then she kills that queen. All the mites and the workers that were in there become hers. And so then they continually get those mites and a lot more mixing of species going on with those social parasite species. And some social parasites are actually hyper social parasites. So that makes it even more crazy.
I find that fascinating because in reality, what you are looking is a microcosmos in front of your eyes.
Mm-hmm.
Something to help all of you is the basic thing is make sure that you clean your area and have a magnifier, or maybe buy a very cheap stereoscope. You can buy very cheap stereoscopes today in less than, I don't know, $40, $30.
USB one.
Good. And be familiar with your environment. In nature, you will not see this often. Number one, we have not been able to study too much, but number two, also the nature balance all these things. The problem that you have is in the moment that you confine your colony, you need to understand what are you doing to try to keep it as natural as you can.
Okay.
Yeah. Okay. Next question.
Sure. What are the different types of mites that would normally show up in a colony, and how can you differentiate them?
Okay, really quick, really fast. Usually when you find hard bodies, kind of caramel color, dark browns, moving around really fast, you are encountering the type of predator mites called mesostigmata. They're free living, moving on. When you see the white ones, kind of transparent, rounded, are called astigmata. White ones usually are, typical, easy phoretic ones. We do not know what the adult does in this case. So maybe
they don't even look like them at all. So linking them together, you have to culture them to know what the adults are versus the phoretic form.
Perfect. And also make sure when you have your colony, not only look your ants, keep your eye where are the larvae of the ants to see if there is anything moving inside with the larvae, right?
Yeah, because they may be riding on the ants and then they get off and go to the larvae.
And the food sources of the ant also can be another ones that were specialized to be there too.
Okay. So that big mite that Zenuri sent that he thought might have been a tick, that is a mesostigmatid mite?
Yeah, it's a mesostigmata.
Yeah, and then if you go down to the later one where there's a whole bunch of little teardrop-shaped ones on the abdomen, those would be the astigmatid mites?
That's right.
Yeah. And then the scudicarrid mites are also really common, and we sent a picture of that. It was a black and white one in the
And that ones are kind of little hairy, really
they
nice.
have really hairy back legs.
Yeah.
And they're brown and kind of turtle looking.
Yeah. Yeah, I called it turtle mites.
Okay, so all right, so I'll just ask this question. I'm not sure if you're going to be able to answer it, but how common are the predatory mites, and what percentage of mites in like a normal enclosure are normally going to be harmful, helpful, or indifferent?
Unfortunately, we don't know. We do not.
No, okay. I think usually the predatory mites are going to be the big guys, though, those mesostigmatid mites. The astigs, not so much. Heterostigs, not so much. Like scudicarrids, no.
Okay.
Right, they're mostly fungivores, Ron.
Yeah, that's right.
But they could, even if they're not actively harming your ant, like the scudicarrids could be carrying a fungus that is pathogenic for the ants.
Yeah.
They have little pockets that they carry fungus with them to colonize their new habitat. So that's a separate risk.
Yeah.
And one thing with mesostigs that sometimes is interesting, you can have 3 different size of mesostigs where the big one eat the next one and the next one eat the smaller one, but the smaller one is able to move to areas where the big one cannot get in, and there is a balance there of predator-prey situation. Whatever it is, you need to observe. You need to observe if the mite that he sees is often running on the colony or often is running on the ant, because also behavior is teaching him if you are him or her, if the mites are associated as a predator or is associated to as a parasitic. Yeah. Oh, interesting. We have balostiums.
Yep. That's a parasite.
Yeah. That's the one when I go anting. That's what I see normally.
That is
red ones.
Yeah, I don't see that one hardly at all. That one's pretty rare,
that
but
one,
distinct.
Yeah, that one is happen when the ant is moving on grass soily or then mulch soil, and there are the erythriids, and they are waiting for somebody to pass by, and that is the larvae. The adult is a free living predator. Here's a very interesting situation with that mite. The larvae is a parasitic, the adult is a predator. That means that he more or less annoy you and later help you. It's a contradiction.
Okay, so would those be parasitoid?
Yes,
would they
parasitic. That ones are parasitic.
Yeah, they're just kind of sucking the blood, but they aren't going to kill them.
Yeah, but if you have too many, it can stress the queen.
Okay. Okay, next question. This one is from Lord of Ants. He asks, mites, Oplidus species, that attach that attach to Campanatus queens and worker legs via the tarsus tibia are some of the most commonly seen examples of mites associated with ants, especially in the eastern United States. What exactly is their relationship with the presumed host? Is it parasitism, myrmecophily? I'm not sure how that's pronounced.
Catherine, that is for you.
So those are Europodid mites. The Europodine mites. It's Oplidus is the genus. I just typed it in there. And yeah, they're super distinct, really big. They look like the ant's carrying little balls on its tibial spurs. They kind of clutch onto that. And I don't think people really know what they're doing. Do you know, Ron? I would think that they're maybe predators or scavengers of nematodes and things.
Or the other possibility, but unfortunately, because we don't have, again, several years ago, we didn't have the technology that we have today, is maybe film what the adult does or the larvae does when it's close to the food sources or the larvae, because maybe that is the clue that we will know what they do.
Yeah, I'm uploading a picture of some pictures I've taken. Oh, sorry, it's not popping up as an actual picture. You got to download it. I don't know. But the one in the top right is Lasius alienus with Oplitis on its tibial spurs, if you wanted to see that. And those ones tend to be really species specific. That the Oplitis species are very specific.
Wow. Okay. So Seraphine asks, there's some sort of hysteria among ant keepers, especially European, that feeder insects are prone to have mites and will infect the ants, so everything has to be boiled or cooked, which you've already spoken to an extent, which makes things a lot harder for the ant. Are there any tangible data on how likely it is that feeder insects will have mites? I assume big corporations would go out of business very quickly if their food kills the tarantulas and mantids of their customers. And this Seraphine specifically has never had feeder insects with mites. And how likely is it that, if present, those mites can actually infest the colony? From what I've a lot of parentheses here. From what I've read, most mites are rather host specific, with the exception of large predatory mites like red velvet mites, whose larvae suck on all sorts of arthropods during the first stages. I think there was a question in there.
Yeah, that was a very long question. I think I was typing at the same time, so I missed it. Sorry, guys.
Let's go to the next one, and I'll summarize that one in a second.
Okay, thanks.
Okay. How do mites affect fungus-growing species like Atta or Acromyrmex? Is there a special kind of mite that takes advantage of their fungus gardens?
Okay, that is what we mentioned before. I mentioned that there are mites that carry their own fungi, go to where the other fungi is. They might carry a parasitic fungi that will parasite the fungi that the ant used to feed. It will kill that fungi, and that is the way that this might survive, eating the fungi that he carries to parasite the fungi that the ant eats.
Yeah.
And that will kill eventually the colony of the ants, right? Because
they're relying on their special kind of farmed fungus, and then the mite might kill them. Yeah.
In other words, there are mites that are farming the fungi. Yeah.
The question was how high are the chances that feeder insects will bring harmful mites into an ant colony?
Like collembolas or something like that?
No, like crickets.
Mealworms.
Yeah, crickets and mealworms. I would think pretty high, especially those rhizoglypha things on, grain.
Yes. Okay. Yeah. Okay. You just open a can of worms. Here is. Rhizoglyphus is a mite that looks like the mold you saw before. And Rhizoglyphus is a genus that is plant parasitic. Is directly important for us to keep them out of this country. There are 50 different species around the world. And unfortunately, when you are collecting plant material to feed your crickets, these mites attach to them and they move around. And that is the reason that quarantine start to show up, because, hey, you are moving up a mite that is harm crops.
And I think with mealworms and things like that, they would have acarus and various grain-associated mites that would get introduced.
Yeah, like the Glyphosaurus. Yeah, Glyphosaurus. That is very nasty. Yeah.
And this is Armando here. Just want to. The signal's really low. Chops that. Let me. There's I believe there's some suspicion now that mealworms are holding a lot of mites like that unknown mouth parts, and that's something researchers are working on to see if they can get those mouth parts on an SEM, just chop their heads off, take a look. So they're not visible on the body at all.
That would be interesting.
So are you saying that even like I run down to PetSmart and buy mealworms, that those are very highly potentially carrying unwanted baggage for my ant colony?
Yes. You have a risk. You do. Yeah. One of the reasons that maybe I specialize raise these food resources in sterile environments to prevent the next step to get contaminated.
Mm-hmm.
But again, basic things. Again, remember, ants are well organized. Try to understand how they are organized. Each of you are very knowledgeable with the ants that you are working on. You are the first person who will notice if something is wrong. The behavior, collect the mites, if there is mites. Perhaps one of the most difficult things to be here is to detect if there is something that is associated between mite, fungi, or mite, nematode. But with the help and doing careful observations, we will start to understand this. Our concern also is, okay, we are talking about species here in the United States, but when we start to import species that we not know anything from other, remember, they will come with their own mites. And who knows which ones are these ones.
Mm-hmm.
So that cool, thing that you brought into your house that came in a nondescript package from China, the opening comes into your house.
Okay.
Yeah.
Yeah.
I have a question which got overlooked, I think. A friend of mine is a beekeeper, and they often spray formic acid on their bees to get off the varroa mite. First thing is, I wondered if acid spraying ants are actually less susceptible to mites, or are the mites on those more specialized so they can deal with the acid? And the second question is, I've heard that some people say that pseudoscorpions, Helifer sp, can be used to combat varroa mites in beehives because they basically eat the mites.
Oh, that's interesting. Your question about whether like Formicine would have more mites than like Myrmecine, for example. The things that make formic acid versus those that don't. And then the dolichotyrrhini have a different kind of chemical, but I'm not sure what it is. I haven't noticed that the Formicine have less. I would say they have more in general than the Myrmecine. But the dolichotyrrhini, I've only looked at a couple species of those, and they were the kind that kind of move their nests a lot, like Taponoma. So they didn't have very many mites, and I don't know if it had anything to do with the chemicals they secrete. But I have thought about the use of formic acid to control the mites, and I think the formic acid is supposed to help with fungi and fungal growth inside the colony. That's why they make it, or it's sort of an antibacterial sort of thing. So potentially it could be used against, like we could, manufacture the formic acid like we do for bees, and then use it to treat the colonies. But I don't have any idea if it would work. And then the thing about the pseudoscorpions is pretty cool. I hadn't heard about that, so that's neat.
I've read a study, I think it was like half a year ago or so. I don't know how old it is, where they discovered that, I think it was Lasius ants, that they're basically using their formic acid to disinfect the new home before they move.
Okay, yeah, that makes sense.
That was pretty interesting.
Just based on the information that you provided for us, I have a question. So if we do notice that a colony has mites and such, you said that having the colony move can significantly decrease or help the colony. Because one thing that has been Dr. Ochoa has mentioned a bunch of times is the confinement is what leads to the really like the mites being able to propagate to the extent that they do. So moving the colony would potentially help a lot.
Yeah, so long as you don't you notice it before the mites are all phoretic on the ants, right? So if you notice there's like a ton of adult stages or something running around in the food and things like that, then you could, pull all the ants out. I mean, it would be a big job, and if they're all coated in mites, that's not going to make a difference at all. So you'd have to cleanse them of mites as you moved them over somehow. Potentially you could kind of scrape it off of them a little bit and at least knock them back, or maybe dipping them in water would knock some of them off, but some of these hold on so tight with little suckers. That like the little ones, I think, would be the hard ones to get rid of. The big ones you could knock off easily.
Yeah. And they attach in the base of the coxas. They attach in the base of the
really tight places where they're not going to get knocked off.
Yeah, that is the reason I said that learning about mites, maybe we'll start to identify which mites are useful. Who knows if with all these observations, suddenly we discover that there are a couple of good predators that are associated feeding on the phoretic mites and find a balance. Like he mentioned the pseudoscorpions. Yes, the pseudoscorpions eat mites. Furthermore, there are pseudoscorpions that live with passalid beetles, and these pseudoscorpions are always under the elytra of the passalid beetles. And what these pseudoscorpions are doing is cleaning and eating the mites that they try to parasite the passalid beetle. And that means that, yes, there is a lot of things that we can learn from this.
Wow. Okay. One of our members asks, are there any easy to acquire natural or artificial mite deterrents, or in general just any way to deter mites from ants? And I feel like you answered this to some extent already. Or Tanger.
Yeah, I've heard about mite paper. What is mite paper? Do you know, Ron?
I don't know.
Like
maybe
chemically treated paper or something that's got some sort of miticide.
Or something sticky that the ant is strong enough to away, but not the mite.
I think it's a chemical, but that was something I was thinking of. But no, I don't actually know if it would work at all. But I've heard about people using it for something else, like maybe for grain storage or something.
Yeah, one of the mites that will give you easy pollen that probably is not associated with the ants at all will be the Thyrophagus protescenti, the mole mite, because it's a perfect contaminant for whatever you do in your home. That keeping that out is help you a lot. Like I said, I see that. And another thing is true. Maybe all these confining scenes that you have are very small, very tiny. That is the reason that it's very important that your queen is clean, because the confiner is so small, it's really easy the mite to run it faster than the ant.
I see.
It looks like that mite paper stuff also kills ants, so not really recommended.
Okay. Zenere, do you want to ask your own question? The one that you have listed in the questions channel?
You can go ahead and just read it.
Sure. Yeah. Okay. He says, as you know, I have been directly involved with mites or ticks on my queens caught during the nuptial flights. My question is, was my course of action in cleaning and removing the mites from my queens indiscriminately the proper action to take? I literally picked them off with tweezers, and in some cases I had to refrigerate the queen in order to get them off while she was being still. Are there other methods of removing mites that might be less harmful to the queen, since improper refrigeration could kill her?
I think that is probably a good method. The physical removal of the mites is really the only guaranteed way. So I don't know.
Yeah, keep your queen under observation. Get a cheap stereoscope or a nice visor with a nice lenses, and look and keep your eye on your queen to see what is going on.
Yeah, and I believe Armando has repeatedly linked a very good and expensive microscope that would be good for that. Correct?
Yeah. By the way, somebody mentioned. Yeah, Drosophila cultures. Drosophila cultures are very susceptible to mite contamination, but also there are several mice that we know that they live with Drosophilas, moving and transfer genetic material from one part to the Drosophila, but that is another story. Anyway.
Okay.
I just linked the microscope again for anyone that cares.
Thank you. Okay, Lord of Ants asks, Ant-associated mites can have very specific attachment sites, or they may not and can be seen all over the body. I've encountered two relatively active species, analogous with specific anatomy, both found on Camponotus. One that attaches under the head, and another that's mostly confined to the propodium. Such mites seem always to hold a low population density in wild colonies. What are the reasons for choosing such sites, and what are the implications for the ants?
Yeah, so like that picture of the ant with the mites, and then like the big mite under the head, and then the mites on the mite, and then the mite on the nematode. That one. Part of what I was doing when I was studying those things was I was looking at where all the different mites were riding on the ants, and they do tend to have very specific places. And so like Anthenophorus, that big mite, will ride on the underside of the head. Once that spot's taken, then one gets on the abdomen on the side, and then it'll balance the load on the other side. So I think they're trying to streamline it to some extent for the host so that they don't get knocked off and they don't hinder their host too much, especially if it's a flying host. You know, you don't want to fall off. So you'll see them kind of like around in the mouth part area. I've seen that on Camponotus, and also between the legs on Camponotus. They'll hold on to that petiole area and tuck themselves down inside between the petiole and the thorax. So I think it's a very species-specific thing, so they can divide up this habitat that's an ant. Some of the astigmata, which are the teardrop-shaped kind of light-colored ones, I see those a lot on the underside of the abdomen. And then also on the legs. If it gets really hugely populous, then they just get on anywhere. And that's when you know you got a real problem, I think. And it may be that they've co-evolved with these ants, so they know the best place to ride on them, and that's something specialized that they've evolved. And then the ones that don't have very specific locations that they ride might be newer associations, or they might not be specific at all to the ants.
Awesome. So Caitlin, in the picture that I just posted with the mite there, on one occasion, I think it was maybe 2 occasions, when I caught these queens after nuptial flight, that mite was actually tucked in under the front legs where it meets the body, kind of like in that fold.
Yeah. Yeah.
And so it was actually under, I mean, when I saw it, I was like, no way. Because all I saw was the butt and a couple of legs poking out. And I was like, oh my gosh, I can't even believe the queen can walk.
Yeah. I find a lot of them like tucked under the coccyx like that, between the back leg coccyx and that little tiny square shaped area, or under the front legs, really tucked in tight. Yeah. It's pretty cool.
Yeah. Yeah, that mite. One thing that I will do with that mite, if you can take that mite out and put it in their own vial.
You have that mite, Ron.
No. But for the next one, this is what I am thinking about. Next time that you collect a mite like that, take it and put it in a small vial with and have the white mites, and look if that might go and prey on the white mites.
Mm-hmm.
That is one thing that you can do. You know, just experiment. Try to figure out
if it's actually feeding while it's on the host. That would be neat.
Yeah, because the body of that mite looks more like a predator mite than a parasitic mite.
Right.
Yeah, I could tell you that when I plucked it off, I don't think that it came off easily. I don't know if it's just because of its claws. But
no, the chelicera. They use the chelicera to attach the hair. The chelicera
right onto a hair.
Yes, that's right. The chelicera of these mites are in a way that is a perfect size to a specific hair of the host, and will grab it. We know a little more of these on beetles because, again, and Kathleen, you know this, Dr. Hans Copeland has been working with these mites associated with beetles, and they found the way that they grab the cheliceras, and depends of the hair and the position on the beetle, you will have different species of these mesostigmatas grabbing with the chelicera, the hair, and in that way are able to ride long distance.
Mm-hmm.
Right. So I think the next question that they have, because our gracious host had to step out for a minute, are Pimotes mites found all over the world?
Yes. Many species.
And next question on here, with the links to predatory mite with the links to predatory mites that we know exist on Amazon, that you can buy. Can either Dr. Ochoa or Caitlin give us a good idea? Set up a mite quarantine for our new queens to disinfect the existing mites in our queens prior to
That is such a cool idea. So you're talking about like having a tube or something that would have these predatory mites in it, and then you would put the queen inside there when she was first introduced. That's a really neat idea. I would love to see that actually tested.
Yes,
so
me too.
Really try it.
Yeah, me too.
Yeah.
And one thing, everybody today is glued with a phone, and you can put a lens over the phone and film what they are doing, because I will love to see that.
Mm-hmm. Do you know, Ron, do you know if the phytosaids, do they hunt by sight or chemical cues or what?
From all the predator mites that I am familiar, I think, in base of what I know, and I has been seeing a lot of them working, I think Neosciurus cucumeris will be a very interesting mite to try, because it's very small, it's fast, it's tiny, and we know that they eat astigmata mites.
Mm-hmm. Yeah, because I'm thinking about the astigmata that might be on these ants. They're just sort of sitting there, not moving at all. Would the mite actually find it as a predator?
Yeah, that is what I am thinking. It would be really nice to try. Don't buy too many. Just buy just one little bottle and try the experiment and see what happens.
So cool,
yeah. That is something
Maybe I'll try that.
Yeah. Yeah, it's really fun.
I have probably 80 to 90 of those C. fragilis queens from nuptial flights, so I'd be definitely willing to test the predatory mite, like quarantine kind of thing with them.
Yeah, those are really big mites, and phytosaids are big mites too, so that'd be like a real showdown.
I think I've read some German journal a long while ago where someone actually did that, and the main problem is that you have to keep your setup pretty moist, because if it's getting too dry, the predatory mites will all just die.
Right. Yeah.
Yes.
They also suggested putting a wet sponge into the outworld, because it will attract a lot of mites, and then you can just put.
Yeah, it makes sense. It does. And one thing that you can do is, when you're using water, try to use sterilized water also, and maybe paper towel, something like that.
So
I actually only water my ants with DI water, so I don't use tap water. I don't use anything because of the chemicals that cities dump in their water, such as chlorine and all that stuff. So
yeah, we can handle a lot more than a little ant could.
So I'm just using tap water, but then in Germany, our tap water is better than the water you can actually buy at the supermarket.
Yeah, distilled water is the purest, though.
Yeah,
I think Kylori wanted to elaborate on the basic setup that he was thinking about.
Please.
All right, so I know that predatory mites are generally going to require an existing food source in order to keep it going. So the idea was to set up this little screw-on cap container with a little bit of moist, like oatmeal or something, and house, pretty much intentionally raise a culture of grain mites, and then introduce the predatory mites to that, so that there is a permanent food source that we can keep on hand at all times for when we go out queen hunting. Mm-hmm. And the idea is to, once we catch a queen, place said queen in the culture, leave her there for like a day or two, give the mites enough time to figure out, hey, there's new food, and see what happens.
Mm-hmm.
How would you be sure that you weren't going to pick up some of the grain mites with your queen when you took her out of there?
Refrigeration, test tube,
you could look at her then.
And refrigeration, give her a good look over.
I think that's pretty cool.
Yeah, when now we are, for example, we are working right now in controlling nematodes with predator mites, and what we do is when you buy the colony of predator mites, they come already with pieces of wood and the Tyrophagos protescenti mite. And the mites are feeding that, and you can put that in small glass, and with a fine brush, you just can pick up the predator mite from the Tyrophagos because it's really easy to separate them. And you put the predators in a clean container with the nematodes, and we look if they are eat or not the nematodes. And that is what we are doing. And it's an easy way to do if you have, small clean containers.
So you could keep your culture and then isolate them as needed.
So yeah, just pull out a few mites into a separate thing that would have the queen.
Yeah.
Huh. Okay, so you're saying that you could get a feeder culture for the predator mites that aren't feeding on like insects and stuff, something that would not potentially be harmful to the queens?
The predator mites, when you buy a predator mite container, it will come with a specific size. Size is tied with a colony of Tyrophagos, this the mole mite, with the predator mite.
Oh, okay.
And what you do, you dump a little bit of that in a clean container. You pick the predator mite that you want to use, put the predator mites in the other container, leaving behind the wood and the Tyrophagos, because you don't want to Tyrophagos either to the clean container. That will take away what you are trying to do. That is, use just the predator mite.
I see. That sounds... I'm going to leave that to other members. That sounds like a good way to just introduce the mole mites to the clean queen.
I mean, the predator mites are really big compared to the Tyrophagos. You probably wouldn't even see the Tyrophagos, but you would see the predator mites running around.
Yeah.
Yeah, you will see something hairy and someone completely smooth. The smooth guy is what you want. Okay. They look like Ferraris running.
Yeah. Okay, I think we just have one last question here, and that was asked by Coltrane, and he asks, just curious, and it's not a question that needs to be answered, but what attracted you to mites? Was it a passion at a younger age, or possibly something you grew into, or was it something that you happened to stumble on and found a love and passion for?
I have a good story. So, when I was an undergrad in college, I was going to be a veterinarian in my future. And then I was in a class with Hans Klompen, who's the person that we talked about that studied Uropodyna. And he said he worked on mites and ticks. And I thought he said mice, M-I-C-E. And so I thought that was kind of veterinary. And so I contacted him for research opportunities. And then it turned out that I really liked mites instead. So that's what I work on now.
For me, oh my God, how I would say, I born in Costa Rica, Central America, with a lot of rainforests in that time. And I remember coming out of the house, and my family was kind of low-key family, and in other words, not too many toys. That means that my toys were insects and lizards. And I became in love with the beetles and all these things that they were around the beetles. And that's it.
Well, okay. Do you guys have any more time for us? I mean, it's been nearly 2 hours, and it's been amazing.
Yeah.
I think my little baby's about to wake up from her nap, so I should probably go.
Okay. And I have visitors that need to take care. But one thing that you can do, as we are in contact, please send me an email with some of the questions. Perhaps Kathleen, also you want a copy, and we can go over and pass it to you guys and address the issues that maybe we didn't cover. And I'm always happy to help, because what we want is that really the goal here is that you keep doing what you love to do, only to be safe.
Yeah, exactly. And we're more than happy to work with you guys in any capacity that you guys like. I mean, just forward any ideas that you have or any way that we can help. And I'm sure the community will be glad to come together to help with you guys.
So, Caitlin, another thing I've been doing, I don't have a lot of time to do it. This has kind of been when I have spare time. These guys have so much good information that it really, really should be a short communication. I think it's Myrmecological News kindly told me to go away.
They're kind of high tier. Yeah.
Yeah, well, I was hoping I could get a short communication out, but in the future, I have a research chat channel in here. When I have time, I'm trying to get certain data sets organized, so you're welcome to join in as well if you want.
That'd be great. Thanks.
Happy to help.
Okay.
All right.
I guess that wraps it up. So that has been Dr. Campbell and Dr. Ochoa. It has been amazing having you guys here. You've given us so much. You've given us so much to talk about and different things to try. It's just been really amazing.
Yeah. And anything that we can do to help you guys as well. You know, I mean, very willing to send in stuff to you guys.
Exactly.
You know, if you guys actually need live specimens to deal with or whatever, you just let us know. I have no problem getting PPQ permits or doing whatever's necessary to help you guys with what you do.
Okay, thank you to all of you, and I would say you are our eyes on this. What you observe and what you record will be very helpful, not only to yourself but to everybody.
Yes, thank you all for the opportunity, and thanks for the really challenging and interesting questions.
Yeah, thank you guys.
Yeah, it's been amazing.
Bye-bye.
One
Bye.
thing I wanted to add is that it's we are people from all over the world, so you don't need to think that it's just the United States, because I'm from Argentina, and there's people from India, there's people from China, everywhere. So yeah, we're willing to help.
More opportunities, yes.
Yeah.
Thank you so much for this.
Thank you guys. Bye.
All right, take care.
Welcome. Bye-bye.
Oh man, that was awesome.
That was good stuff.
Yeah, it was really good. Lots of stuff to think about, especially with the predatory mite setups. That's really, you know, like
I can't believe I stumped the experts.
When we first were setting this up, I talked about the lemon juice stuff, and they kind of just like laughed it off because it just didn't seem. But like, I mean, when I brought it up, it seemed like it's kind of plausible. I mean, because especially like if they're using formic acid to treat mites with bees and stuff, I mean, surely that applies to some degree with ants. Maybe, I don't know. Anyway.
I can't believe Ants Canada was right at something.
I mean that
far.
Well, I mean it wasn't.
Yeah.
That was part of the community well before Ants Canada got a hold of it. But anyway, okay, I'm gonna stop the recording here. Thanks for joining in, everybody. It was really great. It'll be transcribed and up hopefully within a few days.